I'm just going to talk a bit about our last 4 years of measurement at Ashley Dean on carbon and nitrogen inputs and losses under Lucerne. And so back to the concept of this larger MB project, that by increasing carbon inputs to the soil, we can reduce nitrogen losses on farms. We've seen a lot of work from Will on plot scale studies of manipulating carbon, and there's a whole lot of other process work involved in this project that's happened at the small scale. But there's also a big advantage to having measurements of carbon and nitrogen balances at the scale at which farm management is actually applied, so at the paddock scale. And as David mentioned in the introduction, we've been working at Ashley Dean, which was recently converted from a dryland sheep and beef farm to irrigated dairy. And there's this strong perception that irrigation amongst farmers and some researchers as well, that irrigation increases soil carbon. As you can see in the pictures there, you go from this dry brown environment to a nice lush green paddock. Obviously it must be gaining carbon. So Ashley Dean really gave us a platform to use our paddock scale methods for measuring components of both the carbon and nitrogen balance. And we went to sites on the farm that were planted in Lucerne. And so why would we want to measure Lucerne? Well, for one, it's a valuable forage crop that's used pretty extensively, especially in Canterbury. And two, it's got a really deep root system, which could increase carbon inputs to the subsoil and also intercept nitrogen at depth. So David presented this sort of balance of imports and exports from a typical pasture system in his earlier talk. And this is the concept that we took. We used a suite of our paddock scale methods to measure inputs to the system from plant fixation, management inputs from effluent. Our irrigated site did receive effluent, animal excreta and fertilizers, and exports from the system from harvest being really important, leaching, and then gaseous losses. And using all of these to estimate changes in the carbon and nitrogen balance of our system. So just to look at our specific methods that we use, this is an eddy covariance system, and this is a micrometeorological method that measures net ecosystem productivity, basically the balance of carbon fixed by photosynthesis and losses from respiration of the plant and soil. So that gives us two components of our carbon balance in one. For nitrogen, we don't have anything quite so nice as an eddy covariance system, but we can still look at fixation of nitrogen associated with the Lucerne crop by monitoring biomass in the system and then using differences in the stable isotopic signature of nitrogen in the soil versus fixed nitrogen to partition the amount of nitrogen in that Lucerne biomass that's come from fixation versus uptake from the soil. Our management inputs, we can pretty easily estimate our inputs based on sampling and measurements and lab analysis. Harvest, we measure lucerne taken away by harvest, whether it be mechanical or by grazing animals, which come with an associated input to the system as well. And then leaching of both carbon and nitrogen, we can estimate with our array of large lysometers at each of our irrigated and non-irrigated site. We've got 6 2-meter diameter by 1 meter, 1.5 meter depth lysometers, where we can grow deep-rooted lucerne and also simulate management of the site. Okay, so we're working at an irrigated and non-irrigated site. What is the difference? Well, our irrigated site is receiving kind of on the order of 4 to 6 millimeters of irrigation, sometimes including effluent, throughout the season of October through April. And that translates, if you look at the bottom graph on this figure, this is a representation of how much of the soil's water holding capacity is full. So with 0 meaning the soil profile is empty, and 1 meaning the soil profile is full. And so this is the effect of our irrigation. You see in those summer and autumn months that the irrigated site, the soil is always a bit more full, and mission accomplished. That's why we're irrigating. And that translates into increased forage production. At our irrigated site, we get 1 to 2 extra harvests per year, and that equates to a 70% increase in dry matter production, as you'd expect. But what we're also seeing is that irrigation doesn't necessarily consistently increase net CO2 uptake by the system. This is cumulative net ecosystem productivity for each of the 4 years of our study, measured by the eddy covariance system. So that's the balance of photosynthesis and respiration. And we find that sometimes the irrigated site has higher NEP, sometimes the non-irrigated site actually has higher NEP. And despite the huge difference in biomass production. But NEP is just part of the carbon balance. What if we put all the components together? What you see here is the cumulative net carbon balance, including all the components throughout the 4 years. And this typical pattern you see is sort of Lucerne growth. You get gains in carbon in the system, and then when you harvest, you immediately lose that carbon from the system. So you go back to being sort of either neutral or a slight gain over the year. And as you move through the year, you move deeper and deeper, and until at some point you kind of cross the 0 line. And at that point, the system is actually a net loss of carbon. And because we don't have any other substantial reservoirs of carbon that carry on from year to year, we know that this must be a loss from the soil. And what we see is that when you compare irrigated and non-irrigated, the non-irrigated site is typically losing less than the irrigated site. In fact, we have some years at the non-irrigated site where the site actually gains a bit. So it's kind of on the order of plus minus 1 ton of carbon per hectare per year, whereas the irrigated site is losing 1 to 3 tons of carbon per hectare per year. So that's really in contrast to this perception that irrigation is increasing soil carbon, but it's very consistent with national scale studies of paired irrigated, non-irrigated sites, which have shown a lower carbon stock in irrigated systems. In addition to irrigation, we had this gradual change in harvest management of the system, whereby in the first year we had a purely cut and carry system. So all of the at each harvest, all of the biomass is taken away and fed to cows elsewhere. So it's leaving the site. Whereas in our second and third, in subsequent years, the farm increasingly managed the site with direct grazing, which leaves a bit of carbon behind. And what we saw is that in years where the site was grazed only, these are the years where you can, for instance, our third year at our non-irrigated site, these are the years where you have potential to gain carbon. because of that carbon that's left on the paddock rather than exported. Those were our results for carbon, but we've also looked at the balance of the water balance of the system, and why have we done that? But eddy covariance is often very abstract to people, and they wonder, have we missed something, some carbon in our system? Well, that's really tough to evaluate for carbon, but the exact same eddy covariance system can also be used to measure evaporation of water. And we also measure drainage from the soil with our lysometers. And then input, so those are our exports, and input of water is pretty straightforward to measure with tipping buckets. So we have this whole system, and we know if our exports don't add up to our inputs, we have a problem. But with a few fairly explainable exceptions, we had pretty good water balance closure. So this really, and as well, our measurements compared well with a physically based soil hydrological model. So this gives us a lot of confidence that we're not missing something here. We're measuring all the components of our water and by extension, carbon balance. So now moving on to our results for nitrogen from the lysometers. As I said before, irrigation increases biomass production by 70%, but that doesn't necessarily mean that nitrogen fixation by Lucerne is 70% greater as well. Our measurements indicate that at our non-irrigated site, the Lucerne is fixing about 80% of its nitrogen content, and taking up the remainder from the soil. At the irrigated site, which is receiving 70 to 200 kilograms per hectare per year of nitrogen and effluent, that rate of fixation is much lower. They're fixing less than 60%. So as a result, nitrogen fixation at that irrigated site is only 24% greater. But when you add up all of the inputs at each site, from effluent, from grazing inputs, and from fixation, you find that the total nitrogen input at the irrigated site is 60% greater than non-irrigated. Okay, so how does this translate into outputs, exports from the system? Well, these are our 4 years of drainage from our lysometers. So this is just drainage volume, not on the nitrogen yet. And what we find is that as a result of that irrigation and the previous figure showing that that soil water is being kept full, we find that when you start to get that typical winter rainy season, it's falling on an already full soil profile, which causes an increase in drainage volume. So you get 1.2 to 2.5 times more drainage under irrigation. And this immediately has a consequence for leaching, as certain components of organic carbon and organic components of nitrogen have a pretty consistent concentration in leachate. So the more you drain, the more leaching you get of those components. But those really aren't the leaching losses that we're worried about. It's really that mineral nitrogen leaching, the nitrate losses that we're concerned about. So how does that drainage translate into total nitrogen loss from the system? Well, with 1.2 to 2.5 times more drainage under irrigation, we end up with 1.5 to 15 times more nitrogen being lost from our irrigated Lucerne. What's contributing to this loss? Obviously, adding the effluent, the additional nitrogen loss, but we've also found that, as Will said in his talk, timing of drainage is important. And in our second year of the study, we had a situation where, with a wet summer, where we had the soil was draining year-round instead of our typical summer period where we don't get drainage. And in 2 large drainage events occurring in summer, we got 60 kilos of nitrogen lost from the system, more than had been lost in the previous or subsequent year and the remainder of this second year of measurement. So really high losses during that summer period. As well, grazing management is really important to the nitrogen loss. Obviously, the cows are leaving carbon on the paddock, but they're also depositing nitrogen. And though we had a mix management for most of our sites, we had a strong contrast between our first 2 years of at our dryland site, which were primarily cut and carry, and then being converted to a grazing-only management in our third year of measurements. And what that contributed to was a 3 times greater nitrogen loss under grazing relative to cut and carry. And then I should just note that in this 4th year, we converted the site from Lucerne, the irrigated site from Lucerne back to pasture. So those large losses in the fourth season of measurement are probably in part due to that conversion process. So just sort of wrapping up with some conclusions and recommendations for carbon, we're losing on the order of 1 ton of carbon per hectare per year from non-irrigated lucerne under cut and carry management. Under irrigation, we're losing 2 to 3 tons of carbon per hectare per year. Now, grazing can lead to net carbon gain, particularly if the cows are receiving a high rate of supplementation, in which case they're depositing more carbon on the paddock than they would get from just the biomass that they've eaten on site. For nitrogen, non-irrigated lucerne is losing on the order of 7 to 9 kilos of nitrogen per hectare per year. Losses are 3 times greater with grazing. And then irrigated lucerne is losing 39 to 102 kilograms of nitrogen per hectare per year under a mix management. And while inputs to the nitrogen inputs are increased to the system by 60% under irrigation, losses increase by 85%. So we expect that under irrigation, the soil is either gaining less or losing more nitrogen. And again, that sort of agrees with a national scale study of paired sites, which showed that nitrogen stocks were lower under irrigated sites. So we realized that we only get a comparison of 2 sites using our paddock scale measurements because they require quite an investment of time and money. But what can we take away from this single comparison? Well, it looks like lucerne does not represent a mitigation strategy for increasing soil carbon or reducing nitrogen leaching. We're losing soil carbon from these sites, and we're also losing high rates of nitrogen at high rates by leaching. We can use a grazing strategy to decrease our carbon losses, but there's a trade-off in that that will just increase the nitrogen loss. One particular strategy which might help mitigate both carbon and nitrogen losses is that we've identified that our carbon balance oftentimes goes negative at the end of our season in autumn when the lucerne is is lucerne growth slows down, but the soil is still warm, and the conditions are good for microbial decomposition. So reducing irrigation, using a deficit irrigation approach, particularly at this time, could decrease soil carbon losses. At the same time, not having the soil water profile full at the end of the season when the winter rain starts could also prevent additional drainage and potentially minimize leaching. So that's the results of this MB work. And as I said, the Lucerne crop cycle has ended and we've converted to a different pasture cover, but this work really continues through a number of other projects, including an MPI Sustainable Land Management and Climate Change Fund project, looking at nitrogen leaching from diverse pastures. We're contributing EDDI data to the data on carbon exchange from our EDDI site to the Carbon Watch New Zealand program run by NIWA. We've recently instrumented the site with more capability for measuring gaseous nitrogen exchange as part of a New Zealand Agricultural Greenhouse Gas Research Center project. And we're supplying all of our data and working closely with both overseer modelers and then other modeling studies. And I should say that we're also feeding all of this data to Joe Sharp, who's going to speak later, and she's really taken our data and used it to its fullest potential by including it in some modeling scenarios. Right, thanks very much. This recommendation of deficit irrigation technique, and that it makes sense because plants can continue to access water when the soil water profile is well below full. I mean, I think they're usually even half full is probably plenty for plants to continue on. But so what kind of approach could we use to what sort of deficit is sensible? And I don't know of any studies. I mean, I know deficit irrigation is a practice in other, in horticulture, but I can't answer your question. Okay, David's asked about the value of these large lysometers. Why do we need to go to that trouble? Well, I mean, for one, we have lucerne, which I'd said is a deep-rooted species. Typically, the typical lysometer experiment in New Zealand is with 50 centimeter diameter and by, I think, 70 centimeter deep lysometers. And so they're just not going to capture that advantage that the lucerne has of accessing deep water and potentially intercepting nitrogen. So we needed the deep lysometers for the lucerne crop. Further, the inputs to the system are very heterogeneous, particularly from grazing. And there's a lot of work on how urine patches contribute to nitrogen leaching losses. So we wanted to be able to apply inputs to these lysometers in a sort of natural sort of patch size and density. So we needed a reasonably large lysometer to do that. And not just for lucerne, but now we've moved on to looking at more diverse pastures. And so having the larger area is really beneficial when you look at the heterogeneity of these species mixes. So there's a bunch of different reasons that, but mainly we want to be able to integrate the heterogeneity of the paddock environment. Yep.
I had a question, if that's okay. We've got some time, so
yeah, sure.
just getting back to this, I guess this idea about, which you alluded to before about the different, you alluded to that to the trade-off between managing for carbon and nitrogen. And I'm just wanting to, maybe it's for everybody perhaps, get a sense of, you know, what would be the management solution to trying to resolve those two things?
Well,
are you going to focus on nitrogen because it, you know, potentially it goes down the drain and up the spout and has multiple downstream effects, or is there another way of considering it?
yeah, I mean, as far as balancing which one you should manage for, I mean, that's not a question for me. I don't think. But as far as getting an optimal balance, I mean, that's really why we've collected this data, so we can pass it on to people like Joe, who's using the AppSim model, and she can take our data, parameterize the model, and then we only got one set of conditions at our site. What you want to represent is this spectrum of management scenarios to figure out which one is optimal for your, whatever your goal is, whether it be carbon or nitrogen. So, yeah.
Great answer. Sort of both kicking it down the road and giving it a bit of an answer as well. I did have one more, which I guess relates back to the whole question of the soils and how transferable this is to other places. I mean, what you're proposing in terms of the real problem with drainage is this a medium to worst case scenario relative to other soil types, or is that, you know, and how would you go about trying to assess that? Does it require experimentation more broadly? Or?
Yeah, I mean, I think again with the paddock scale work, you really only get at best a paired site like we've got. You don't really get the whole, you can't just put them up everywhere. So yeah, we sort of expect from all these small lysometer experiments that Canterbury is probably actually a pretty bad scenario as far as both carbon and nitrogen losses. But I mean, our site is fairly representative of dairy conversions in the region, so I think it's representative for our region as far as the heavier soils. I mean, I think that, yeah, you have to resort to some of the more replicated small lysometer studies, and then sort of survey measurements like Paul Mudge does.